You often hear this factoid about water. People say that every water molecule on our planet has passed through the gut of a dinosaur. Now, the fact is obviously designed to shock us. I mean, oh my god, are we drinking dinosaur pee? But it also got me thinking, I suppose we could kind of say the same thing about soil. The soil that we farm, plant and walk on today. It was once the meaty leg of a dinosaur, or the branch of a carboniferous lepidodendron, or the lunch of a Permian megaherbivore. Sod, muck, turf, earth, soil. Soil is life. We take it for granted that you can stick a seed in it and watch a plant grow. But what is soil, and where does it come from? We've had several requests to do a podcast on the formation of the first soils on Earth, so I'm really pleased to be able to bring this episode to you, and thank you all for your suggestions. Do keep them coming in. In today's episode, I'll be dishing the dirt on fossil soils with Dr. Ria Mitchell, a researcher at the Department of Materials Science and Engineering at the University of Sheffield. Dr. Mitchell has held postdoctoral research positions at Swansea University and the Natural History Museum in London, where she's worked to understand the relationship between soil and the first terrestrial life on Earth. She employs methods that include computed tomography, microscopy, and chemical analyses to understand ancient soil processes, relationships, and planetary cycles. I'm going to talk to her today about her work and what we know about the formation of the first soils. Where did they come from and how did they form? How do soils fossilise, and why don't they just turn into ordinary rock? Hopefully, we can answer some of these questions and many more. Ria, it's great to speak to you today about soil science. Welcome to Paleocast.
Oh, thank you. It's nice to be talking about my work.
So I like to start interviews by just asking my guests to talk about themselves. Could you introduce who you are and a little bit about your research?
Yeah, sure. So I'm currently an experimental officer in X-ray CT at the University of Sheffield, but I have quite broad research interests. The main one being that I'm really interested in the way in which early terrestrial plants and microbes contribute to weathering and soil development. And specifically, I'm interested in how this occurs on a nano to a micro scale to inform larger scale processes such as biogeochemical cycling. And I do this by looking at modern analogues of these really ancient organisms. So I have a particular interest in trying to solve some of these problems through using novel imaging and analytical tools. So things like 3D imaging through X-ray micro CT and X-ray microscopy, but also things like the synchrotron. And then blending this information and this data together with other information through a correlative microscopy approach. So being able to tie together things like imaging and chemistry and crystallography all together to paint a more complete picture of what is going on. So some of the specific time periods I'm interested in are the Proterozoic, so when we get the first organisms into the terrestrial environment, and also the early Paleozoic, so when we start to get the first land plants.
So in terms of millions of years ago, what sort of time period are we looking at?
So my original research for my PhD was in the Mesoproterozoic, so this was about 1.1 billion years ago, which is obviously quite a long time ago. And the earliest land plants is around, well, it's quite controversial, so it can be anywhere really between about 500 and, say, 450 million years ago. So again, still quite a long time, but much more recent compared to the Mesoproterozoic work.
Yeah, that's a big time period that you're covering there for sure.
Yeah.
I'd like to start with the absolute basics here. So I know it's in our gardens and it coats the landscape, but what is soil?
Yeah, so it's a really good question, really, because depending on who you speak to, they'll have a different idea of what a soil actually is. So to me, at its simplest form, it is a layer of mixed components. So this could include things like mineral grains, dead or decaying organic matter. We can have things like water in there as well as various other liquids, gases which get trapped in the pore network within the soil itself, and also, which most people will think when they think of a soil, is a biosphere, so various different organisms. Soils, at the modern day anyway, they generally form under 5 different factors. So it depends on their parent material. It depends on whether they've got a biota or not. The time that it takes for them to actually form, the relief of the environment in which they're forming as well. So the topography, so whether it's high elevation or low elevation, and also the climate can have a huge impact on how soils actually form as well. So I think an important distinction to make between soils as we are familiar with them today is that in the past they didn't always look like this. Today they look the way they do mainly because they have a huge amount of organic material and a biota associated with them. But in the geologic past they look much more simple than this. So whether we still call those very thin mineral horizons that have come from weathered parent material or weathered bedrock, whether we can still call that a soil if it doesn't necessarily have as much of a biological component, is quite a hot topic, I suppose. Sometimes this can be called a regolith. So I know, for instance, when people talk about the sedimentary layer of surface material that's on, say, the moon, they sometimes call that a regolith, but then they also sometimes call it a soil. So there needs to be some sort of a distinction there.
Yeah, I wouldn't have thought soil on the moon. That doesn't seem,
exactly.
to be right.
Yeah, it's
So there's obviously a huge difference between soils in the past and now, but soil is really important, obviously, for life on Earth today. And you touched on a few things that are in soil. So what is it that makes it so important? Why do we need it, basically?
Yeah, so soil is it's one of the rare parts of the planet where it's at the interface between the biosphere, the atmosphere, the lithosphere, and the hydrosphere. So it kind of impacts all of those different spheres, but also all of those different components it impact the soil as well.
So by each of those terms, you're kind of meaning basically like the water cycle, the rock cycle, the living things.
Yeah. So they sort of link together, and they both affect each other, if you like. But in terms of why it's important, I mean, for us humans, obviously it produces food for us to eat, so crops and whatnot. And then in turn from that, it feeds the animals that some of us eat. There are also environmental aspects for why soil is important. So it contributes to things like stabilization. So if you've got a lot of biology, a lot of plants growing on the surface of a soil, then that can stabilize environments, which contributes towards preventing things like flooding, and also enables soils to be able to hold water as well. So a lot of that water that you see in the floods, for instance, in this country that we've had over the past month or so, the more soils we have and the better formed these soils are, then they have this ability to hold more water. They also are important for many other reasons because they provide a home for various different microbes and microorganisms, but also a home for us. So if we didn't have soil, we wouldn't have trees, and then we wouldn't have wood as a building material, for instance. And the soils, which contain a lot of carbon, so for instance, peat bogs, they contribute hugely to the atmospheric carbon cycle. So peat bogs are a really, really good example for holding lots of organic material, which enables reduced carbon and reduced carbon dioxide in the atmosphere. So that contributes towards a larger scale cycle, such as climate cycles. So there's a whole different range of different reasons why soils are considered important in there. But I think that touches on the most important ones.
Yeah. It sounds from what you're saying, like you're basically saying that we couldn't really have a planet without soil. A planet Earth, as it were.
Yeah,
I mean, obviously you can have a ball of rock, but a living planet.
Yeah, this is it. I think if we didn't have them, or if they weren't as almost mature as they are at the modern day, then it wouldn't be very habitable at all. Not least because we wouldn't have food to eat and we wouldn't have crops, but also, as I mentioned, things like climate change. So the ability for soils to regulate the carbon cycle as well. So yeah, they're very, very important.
So let's talk a little bit about how soil actually forms. And I mean, like in the very beginning. I want to kind of paint a picture of the ancient Earth in the time period that you're talking about, from not just millions of years ago, billions of years ago. What was it like before there were any soils? Would it just have been bare rock with, some areas of sand and things in it? What might we have seen?
Yeah, so I think it would have been a lot like that. A lot of the reconstructions you see of the sort of Archean and Proterozoic Earth is very barren with a lot of volcanoes in the background, it seems to be. But so you would have had things like lava flows, which would have been weathered in a way. And if we're thinking about a time before biology was actually around, then especially in the terrestrial environment, then these rocks may have been weathered by abiotic processes. So you might have had chemical weathering occurring through what are called hydrolysis reactions. And this is just essentially the way in which carbon dioxide can get into or be connected with the rainwater, which is coming from clouds in the sky, and it can permeate into rock surfaces and actually dissolve the rocks apart, which means that we get lots of grit and dust and smaller scale material, which can then be washed away by rivers and then can be deposited elsewhere. So I think we'd have a mix of bare rock surfaces, and then we'd have these almost regolith, this loose sediment on the surface of the Earth.
And at what point do we start to see the first life on land? You were very specifically saying there terrestrial life. I'm assuming there's quite a big difference between looking at the formation of soil and looking at, for example, the formation of sediments on the ocean bed. I'm assuming you don't get marine soil.
No, that's true. So we start to get the first truly terrestrial organisms in the Mesozoic, the Proterozoic. So this is going from about 1 billion years ago back to about 1.5 billion years ago. So before that, we have organisms which are cyanobacterial mounds called stromatolites. So we know that these existed up to around about 3 billion, possibly even 3.5 billion years ago. So we know that there was life on Earth before we start to see the first terrestrial life. But these stromatolites tend to only really live in marginal marine environments. So they had to be constantly wet, essentially. So they weren't really living in a truly terrestrial environment. So there are numerous examples from the fossil record of truly terrestrial crusts, microbial crusts in the Mesoproterozoic, so around about 1 billion to 1.5 billion years ago. And some of these organisms are thought to be created by cyanobacteria. So these are photosynthesizing organisms that probably formed filamentous structures, and these likely would have colonized the surface of various different sediments, possibly even living as well beneath the surface. And then they would have almost started to aggregate sediment surfaces and start to create these, turning regolith, this loose sediment material, into sort of like a proto-soil, so an initial soil, if you like to use that word. And then later on in the geologic past, we would have had true soils once we get more advanced life forms, such as early land plants, and then eventually by the late Devonian, things like trees evolving.
One of the things you said there made me think of another question about modern soils. It relates to what you were just describing. How deep is soil? How far down does it tend to go?
Oh gosh, so a modern soil can be meters deep. It varies on those factors that I mentioned previously. So for instance, if you've got a soil that's been forming for thousands, tens of thousands, possibly even hundreds of thousands of years, then these soils are going to be incredibly deep and rich. I imagine maybe an example could be something like what's growing in the Amazon rainforest, for instance, where it's constantly had this forest growing on top of it for thousands of years, and it's had the time to mature and the time to develop. One of the problems with these early soils or these early proto-soils would have been that unless they had a lot of organic material living on the surface, then they wouldn't have been able to stabilize very well. So say a storm, say they were living near a river, for instance, and a storm came through and a flood washed over this surface where this proto-soil was developing. Unless it was aggregated enough, that would just get completely washed away. So one of the main and one of the early sort of instigating things that needs to happen when you start to get this soil development is that it is stabilized for a decent amount of time to enable those soil forming processes to actually start occurring.
You mentioned a couple of habitats that you might, or areas that you might see one of these early soils. Were they confined to particular parts of the world, or could you find them pretty much anywhere?
Well, this is a good question, because this is something that we don't really understand at the moment. So there are various different examples in the sedimentological or the fossil record of what are called paleosols. So this is essentially a fossil soil that has been preserved within the rock. And there are examples of these going all the way back to the Archean. So I think the oldest is around about 2.7 billion years ago, potentially.
Oh, wow.
Yeah, it's some of them are very, very old. But what we don't really understand with a lot of these really old paleosols is how much they were formed by abiotic processes and how much they were formed by biotic processes. So trying to make that distinction, in my eyes, is something that's really important when trying to understand these fossil soils in the fossil record.
Yeah, I was going to ask you about that. I was reading a little about paleosols, and I was trying to get my head around, when people talk about fossils, of course, we're usually thinking about invertebrate or vertebrate animals, essentially, or plants.
Yeah.
So how can a soil become a fossil?
Yeah, so it's essentially the same process as a sandstone or a mudstone that has also been preserved. So it's almost frozen in time, where it goes through the rock forming process or the diagenesis process, and it actually does turn to rock. So it's not as if we have a nice sort of loose soil profile preserved in a rock section. A lot of the time when we have paleosols preserved, it's when there's been a very instantaneous event has happened following it. So, for instance, you quite often get paleosols preserved that are formed on top of, say, a lava flow, and then another lava flow will come in during the soil's formation, will go over the surface of the paleosol and will almost preserve it intact. And then the lava and the paleosols will both go through this normal sort of diagenetic process where they eventually turn to rock. So you do get them in the sedimentological and the fossil record, but they're difficult to find and they're difficult to interpret in the older sediments.
I'd like to come back to how you interpret these in just a moment. But I have another question about some of these really early ground covers and soils that you were talking about. One of them I read in some of your papers and other things was something called cryptogamic. Is that pronounced right? Cryptogamic ground cover? So, could you explain what is cryptogamic ground cover, and do we have it in the modern world?
Yeah, of course. So as I mentioned before, a lot of my research is looking at modern analogues of some of these early covers, and we do get them at the modern day. So essentially, a cryptogamic ground cover is a biological ground cover which encompasses a variety of cryptogamic organisms. So these can be a mixture of early plants, of symbiotic organisms, and of various different microbes. So a cryptogamic organism is a plant or an organism that reproduces only by spores. And the cryptogams are organisms including the bryophyte plants. So these are the mosses, the liverworts, and the hornworts. So I think a lot of people out of the three of those are probably most familiar with mosses, but there are two others as well called the liverworts and the hornworts. Other cryptogamic organisms include lichens, bacteria, algae, and fungi. So at the modern day, we have a variety of these sorts of cryptogamic ground covers that include these organisms, but not always all of the organisms. So sometimes you can have a cryptogamic ground cover that's just say mosses and lichens. You can have some that are liverworts and lichens, some that are just liverworts. So they can be quite varied. And probably most people are quite familiar with them being called a biological soil crust is another term for them. But to me, calling them just a biological soil crust isn't specific enough. So I like to use the term cryptogamic ground cover because it's specific to cryptogamic organisms, basically. So that's essentially what a cryptogamic ground cover is, where you have this surface covering of this cryptogamic biological material, which aggregates the top few millimeters or centimeters of a sediment surface, and then you've got this sort of loose regolith beneath.
So what you're describing there, I can see that is really quite varied. For it to go from being, so obviously we have lichens, we have mosses, like all around us in the woodlands and things. So I'm assuming that this is a ground cover where you really only have those things and you don't have the other kind of plants that make, for example, a woodland floor or something like that. Is that correct? Yeah,
this is true. So the ones that I've studied, I've tried to make them as analogous to the early land plants as possible. So all of these organisms that we get at the modern day are evolutionary, very ancient. So I've deliberately tried to hunt out these sorts of cryptogamic ground covers that grow in these very primitive environments that will only have these sorts of organisms and no higher plants, no vascular plants, if you like.
So where have you been doing that?
Yeah, so a couple of the places we've been to are Iceland, because it's relatively new ground material. You know, most of the lava flows and the ground eruptive material found on the ground surface there, you have an idea of how old it is, like when the last eruption was and that sort of thing. So then we can almost predict how long that cryptogam ground cover has been growing in that particular area. And also the material there is also quite virgin as well, so the mineralogical material. So we can get a really good idea of how these cryptogamic organisms are interacting with the mineral sediment, this mineral regolith, and what it's doing to it and how it's managing to stabilize it and hold it in place and contribute towards these proto-soil developments.
I imagine that this, you keep talking about the way it binds together the soil and keeps it together. I imagine that's really a prerequisite for more complex plants and things to be able to get a foothold then in our early evolutionary history.
That's exactly it, yeah. So at the modern day, they're very much a pioneer species. So they're generally, these cryptogam organisms are the first to come in and colonize something. And, you know, you're wandering around Iceland and you see mosses. It's just seas. It's just got these lava fields and it's just this sea of green where these moss cushions have come in and just colonized. There's a couple of differences with the organisms we find at the modern day and what we might expect to see in the past, though. So we obviously wouldn't encompass all of these different cryptogam components because each of these organisms evolved at different times through the geologic past. So some of the most ancient plants are called cryptophytes, which were generally upright and leafless, so they don't share all of the same characteristics as what our modern organisms do. So, for instance, the liverworts and the hornworts form these flattened thalli, which kind of grow oppressed to the sediment surface, whereas something like a moss, as we can all probably picture in our heads, grows upright and it's quite leafy. So these extinct cryptophytes, they were upright and leafless, but they did have similar anatomical features to some of the modern examples. So, for instance, they have a set of rooting system, which aren't true roots. So true roots, as in, say, a vascular plant, are multicellular, whereas in modern liverworts and hornworts, they have this suite of what are called rhizoids, which are unicellular. So they're a lot more finer and smaller than what the roots are in, say, a vascular plant. But these roots also, and these plants also have symbiotic associations with fungi and or cyanobacteria. And the examples in the fossil record have that as well. So there are, even though the some of the features might not be the same as the modern examples, a lot of the features also are a lot like the fossil examples.
In terms of the modern world, what kind of an ecosystem might a cryptogamic ground cover support?
So we have cryptogams growing in a variety of different areas in the modern world. As I mentioned, they're usually the first colonizers of bare landscapes, and they can grow at high, low latitudes. They can grow in polar tundra to deserts. They often grow in quite extreme environments as well, so quite arid or high, low temperatures and high, low altitudes. So I gave Iceland as one of the examples of where we've been looking at these organisms, and they tend to grow in quite extreme environments as well that other plants potentially couldn't live in, so such as hot springs that have, obviously they're very hot. They have a lot of different minerals associated with them as well, some quite harsh metallic materials in the soil, so they can't. So other organisms couldn't grow, but then the cryptogam organisms are quite tolerant, so they're able to grow in these sorts of environments. So you pretty much get them growing everywhere, to be quite honest with you. I mean, I've seen pictures before of people having moss growing on their cars. It's just, they just get everywhere.
Yes, I imagined when I was reading about cryptogamic ground cover, I was always imagining something quite hard and dry and sort of extreme.
Yeah.
But as you say, actually, when you start looking at the incredible range of organisms it includes, you can be talking about quite a range of different environments there.
Yeah.
And it's interesting from what you're saying there as well, that it seems like, because we of course know about things like ecological succession, and so you imagine that things are always going to get replaced with something else.
Yeah.
But it sounds like these are basically as far as it's going to go in certain environments. They're not only the pioneers, but nothing else is really going to replace them if it's quite extreme like that.
Yeah, precisely. So they're quite hardy little beings. They're quite amazing, really.
And beautiful too. I absolutely love really close-up pictures of these different, especially lichens. Their structures are gorgeous.
Yeah, I think a lot of people see them as a nuisance and a bit of a weed. If they're growing on, I don't know, your fence or in your garden, they might be considered a bit of a pain. But actually, you get down and you look at them. See, this is what one of the things with research I'm really interested in is when you start looking at things at the smaller and smaller and the micro and micro scale, then you start really appreciating how amazing they really are.
Now, looking back again at the first ground covers you talked about way back, how does the evolution of this first cryptogamic ground cover connect with changes that are taking place in the Earth's atmosphere and climate? Is there a connection?
There is, yeah. So basically, these organisms, there's been a lot of research in the past decade or so, which has looked at how, when we start to see what some researchers call the greening of the Earth in the early Paleozoic, when we start to get these first land plants, how that has contributed towards various different paleoecological and paleoenvironmental and then paleoatmospheric changes as well. So a lot of the research has been looking at the timing of when these organisms came into the terrestrial realm and other larger scale processes that were occurring. So one of the biggest themes with the evolution of early land plants is this drawdown in carbon dioxide that we get at around about the same time. So a lot of researchers think that this is because as we start to get the greening of the Earth, we start to get this soil or protosoil development, and then we start to get weathering occurring by these organisms, that contributes towards this carbon dioxide drawdown. And then there are some other researchers as well who think that potentially this drawdown, which would make the Earth a colder place, has contributed towards the Ordovician glaciation. So, what's going on at the microscale within these ancient versions of these cryptogamic ground covers could have contributed towards much larger scale Earth systems. The fact that they were photosynthesizing as well obviously means that they were pumping out a lot more oxygen into the atmosphere. So that could have then, set the way for oxygen breathing animals and could have made their life a lot more easier if there was more oxygen in the atmosphere in the first place to enable them to thrive. So they could have contributed to all of these much larger scale processes.
Right, so we've kind of been talking a lot about these very early ones, these sort of hard crusts almost on the Earth. So take me forward a bit in time then. At what point do we get to something that we would, most of us, consider to be a sort of recognisable soil, something that looks like the sort of thing you buy in a bag from your garden centre? How long does that take to form?
Yeah, that's an excellent question. So again, I think this takes me back to the idea of, what different people consider a soil versus a regolith versus a protosoil. So if we're thinking about a soil as in what we're potentially seeing today, then this would probably come into force around about the late Devonian, when we start to get the evolution of vascular plants and things like trees. So there are a variety of Paleosoils, so these fossil soils again, which have been preserved in the rock record towards the end of the Devonian and then into the Carboniferous, where we get these really well-structured Paleosoils with horizonation, we call it. So the way in which soils are able to form different distinct horizons, and then they basically just means that they form in a much more modern, if you like, way. So I think it's quite a relatively quick change through that period of the Paleozoic. So once we start to get trees in the fossil record, then that's when we start to get more modern soil, if you like.
That's quite an incredible gap, though. You're talking about the sort of start of this, the formation of this, like 2 billion years ago, taking so long for it to get to the point where it's something kind of richer and more organic. Yeah. That's quite interesting, isn't it?
It is, yeah. And I think once we start to get these early plants in the earlier Paleozoic. So it depends on which evidence you're looking at, because the fossil record says that we probably had the earliest plants in the mid-Ordovician-ish, so around about 450 million years ago. But then there's some molecular information that says that they might be a little bit older than that, so they could even have come about in around about 500 million years ago. So then going through then to the late Devonian, when we start to get the trees, that's going on, say, 400 to 390 to 380 million years ago. So it sounds like a long time, but I think in geological time, that actually isn't that long.
Yeah, well, it's the funny thing, isn't it? When we're talking in such huge timescales, we've got a slightly bizarre sense of what is a short amount of time and what's a long time.
Very, very strange.
I'd like to come back to something that you mentioned when you introduced yourself, and I'm really interested in hearing more about, which is the way that you're using various analytical tools, including micro-CT scanning and chemical analyses. So I guess I've got kind of two main questions. First is I use micro-CT, but I'm usually, of course, using it on bones. So quite clear how that's going to work. So how do you use that and apply it to the fossil record of soils? What are you looking for?
Yeah, so this, it's mostly related to the modern analogues that I've been working on, so these modern examples of cryptogamic ground covers. What I've been focusing on lately is looking at the structure of these cryptogamic ground covers beneath cryptogamic ground covers, that is such a mouthful to say every time, that develop beneath different plant elements of the covers themselves. So for instance, looking at the structural difference between one that's dominated by a liverwort, one that's dominated by mosses, one that's dominated by lichens, that sort of thing. So then if we can take sections through these covers, look at the way in which the organisms on the surface interact with the regolith beneath, and then how that structure and that thickness of that soil actually changes as you look at the structure beneath different organisms. That can give us a bit of an indication of how these soils may have changed through time. So it comes back to your last question, really, of how this change actually occurred over, millions or tens of millions or hundreds of millions of years timescales. So that can, even though we're looking at modern analogues, that can still give us a little bit of an indication of how these early land plants and these other organisms like lichens, which have very similar morphological properties, might have contributed towards this evolution of soils through time. So we've had a paper accepted about this, and it should be coming out within the next few weeks, but it isn't quite out just yet. So keep a lookout for that. But that is using X-ray micro CT to look at that soil structure, and to also look at the micro to nanoscale interactions between these surface plants and surface organisms, and how they actually interact with the grains of soil themselves. So, because there's lots of things we're looking at there, so that can contribute towards how these organisms actually weather their soils to sort of fast track the soil weathering process, and how they actually stabilize the sediment surfaces as well. So how much they contribute towards that stabilization and that weathering all in one go, essentially. So CT is an excellent way of being able to do that, because you can take a sample from the field, you can stabilize it and make sure it stays intact, and then you can put it in a CT scanner and look at that structure in 3D. So, it's all very well and good being able to look. The analogy I like to give is that of a Kinder egg. So you can look at a Kinder egg, you can look at the outside of the egg, but you don't know what toy is on the inside. But then you can CT scan it, and then obviously you can see what's going on the inside and figure out what the toy is. So CT has been invaluable for those sorts of studies.
Well, congratulations on that paper.
Thank you.
I'm looking forward to seeing that. If it's out by the time this episode goes out, we'll include a link in the bio. And if not, then our listeners, I'm sure, will keep an eye out for it. Well, another thing you mentioned was the different chemicals that might be preserved. I mean, do detectable chemicals preserve in fossil paleosols?
Yes, so there are various different chemical analyses you can do with paleosols, and I've done some of these in my previous work through multiple different geochemical techniques in the lab. A lot of them include having a soil profile. So this is some work I've done for both my PhD and for my master's, actually, where I was looking at paleosols on the Isle of Wight across the Eocene and Oligocene boundary. So we went to a variety of different field sites. We identified a few different what we thought were paleosol types, and then you take samples through different lengths, or different depths, I should say, of the profile itself. And then you can do chemical analysis and look at the the chemistry or the chemical change through the profile of the paleosols themselves. So those particular paleosols were a really good example, because we had a lot of clay formation, and the clay generally changes in location as you go through different paleosol profiles, and then so does the chemistry. So we can tell quite a lot from the chemistry of the paleosols themselves. There are various different what are called molecular weathering ratios that you can apply. So this basically then tells you how weathered your paleosol is, which can indicate things like time of formation, or hopefully at some point it might help us give an indication of whether any biology was involved. So whether any biology was sort of fast forwarding or fast tracking that weathering process. But that's still on the cards to to try and sort out at the moment.
Right, well, I'd like to switch direction a little bit.
Okay.
And I'd like to talk about fungus, if that's all right with you. I'm a bit mad about mushrooms. I'm actually growing my own mushrooms at the moment, edible ones, which has been an absolute joy. I mean, it's a pure layman's interest. I have no expertise whatsoever, but I'm particularly fascinated about mycorrhizal fungus, and how it lives in harmony with plants, as it were. Could you maybe tell our listeners a little bit about mycorrhizal fungus? First of all, what it is, but then perhaps what relevance it has to your discipline and your research.
Yeah, so essentially, a mycorrhizal fungus is a type of fungus that forms what's called a symbiotic association with a plant, so specifically with a plant's rooting system. So generally, there are two main types of mycorrhizal fungus. There's the first type, which is called arbuscular mycorrhizal fungi, and these basically colonize intracellularly in the root tissue. So the plant lets the fungus get in there and colonize the cell system within the plant itself. So this type of mycorrhizal fungus is found in around about 80% of all modern plants. So modern plants rely on this method of symbiotic relationship heavily. And then the second type of mycorrhizal fungus is called ectomycorrhizal fungi. So you don't need to worry about these two names. It's just important to know really that there's two different types. And rather than getting inside the cells, these colonize extracellularly, so extracellular to the root system. And these only form in around about 10% of modern plant families, but this includes things like woody plants, so trees and similar organisms. So these relationships, these what are called symbiotic relationships, usually form because they are what's called mutualistic. So they benefit both the host plant and the fungus itself. So they both get something from this relationship. Essentially, the plant delivers energy and sugars to the fungus, which it may not be able to get if it was not symbiotic. And then the fungus is able to extend itself within the soil and to extract various different nutrients that it will find within the soil itself. And then it provides that to the host plant. So you can see there's this symbiotic exchange. They both suit each other, so they just sort of get on with it. So that's, it's beneficial for plants to form this symbiotic relationship. But not all of them do, but most of them do at the modern day.
Can they live without it if so many of them have it?
Yeah, they can, yeah. So one good example actually is mosses. They don't form a mycorrhizal relationship with any fungi at all.
Oh, really?
Yeah, so the theory behind that is that so mosses don't have a mycorrhizal relationship with fungi, but another bryophyte called liverworts actually do. And the thought is that because liverworts don't have a true rooting system, they just have this suite of rhizoids, which I covered before, that isn't good enough to be able to extract nutrients from the soil. So they form this relationship with the fungi to help with this nutrient acquirement. But the thought is with the mosses is that they have slightly more substantial rooting system. They're longer, they're thicker, and they're more or less on the same morphological shape and size as what a fungal hyphae would be. So that's the theory, is that mosses don't really need it because their rhizoids or their rooting system are good enough without them.
So when I'm like digging around in the soil in the garden or wherever, and I can see sort of white filaments and things through the soil, is that likely to be the mycorrhizae, or is that just maybe fungus in general?
It's probably a bit of both, to be honest, because various different plants have, what I'd call a more substantial rooting system, but then they might have root hairs, and then it might be that the fungus is attached to the root hairs, for instance. So, and then there's obviously other organisms living in the soil anyway that aren't connected to the symbiotic relationship at all. So it's probably a mixture, but it is incredible actually when you see some of the documentaries about this kind of thing, how far into the soil these mycorrhizal networks can actually extend. It's quite mind-blowing.
Oh, really? Like, what sort of? Are you talking about depth?
Yeah.
Or?
Oh gosh, I don't know off the top of my head, but I've seen various things before where they have statistics about how far that would wrap around the Earth and that kind of thing. But it's a huge amount.
So that would suggest to me that this relationship must go really far back in deep time.
Yeah, it does. So some of the organisms that I've been looking at, I mentioned that the liverworts form mycorrhizal symbiosis with various different fungal types. They have both of the different fungal types as well. So not all liverworts will have the same mycorrhizal relationship. It does vary between different genera and different species. And then some in the modern examples actually form symbiotic associations, but not through the roots. So it can be directly through the thalli. And then some liverworts just don't form symbiotic associations at all. So it's a little bit all over the place. But in terms of how this relates to the fossil record, we do find these relationships in the fossil record, so in different fossil plants. So, for instance, arbuscular mycorrhizal fungi go back to around about 450 million years ago-ish, I think. So it's around about the Silurian to Ordovician. That is from fossil evidence. And then there's various different phylogenetic and molecular data as well, which suggests that the symbiotic associations between plants might go even further back still. So we know that at the dawn of land plants that we probably did have this symbiotic association with fungi. And one of the really, really good units that a lot of information has been found out about this relationship is something called the Rhynie chert. So this is a exceptionally preserved geothermal wetland from Scotland, which is dated at around about 407 million years old. So that's early Devonian. And essentially, it was a hot spring environment, much like the previous example I talked about with Iceland. But all of these plants and various different other organisms that were growing there have all been frozen in time because the waters in which the hot spring was surrounded by was silicified. So it was silica-rich water, which then froze all of these different organisms and the plants in space and time as they were. And the fossils are incredible from it. They're really, really great. But we get a lot of examples from that of these mycorrhizal fungi and various other symbiotic relationships with fungi forming in some of those land plants. So it certainly goes back at least 407 million years. And it probably really, really helped these very primitive land plants, well, I was going to say get off the ground, but that's not the right term at all, to started, let's say, in trying to acquire nutrients from the soils and trying to establish themselves, basically.
It's really incredible. I had no idea you could tell so much about soil from the fossil record. Of course, I knew about the Rhynie chert, as you can imagine, being from Scotland. It's a really fantastic site and fantastic fossils. But it's just incredible hearing what you can tell. I guess it makes me wonder, as sort of my final question is, what is it that we still don't know? Where are the big questions in your research?
Yeah, so one of the aspects that I'm really interested in is trying, as I've said a few times actually, is trying to look at these things on a smaller scale than potentially what we're used to looking at them at. So many researchers, including myself, have discovered and described various different Paleocell soil profiles through the geologic past. But these are always quite thick, they're tens of centimetres to metres in thickness. Whereas I think potentially for these sorts of cryptogamic ground cover analogy soils in the geologic past, we need to better understand the structure at the smaller scale, because I don't think that a lot of these were very, very thick soils and very thick profiles. I think they were literally just surface coverings that were being held in place by this very thin layer of organic material. One of the other elements as well, which is kind of ongoing, and there's constantly research coming out about this all the time, is just the timing of when all of this happened. So again, I cover this a little bit in the paper, which is hopefully coming out in the next couple of weeks, but the discrepancy between what we find in the fossil record and also the molecular or the phylogenetic information. So those dates don't always match up, where the molecular information potentially says that these organisms were living much longer ago than where we find them in the fossil record. So maybe trying to use other proxies and other information to try and establish when these organisms were growing, in addition to the molecular and the fossil information, could be really, really helpful for trying to figure out when these little beasts were alive.
Oh, that sounds really exciting. I'm looking forward to seeing what's in your paper and what's in future papers. This is such an exciting area of research.
Yeah, it's really interesting, and it's so interdisciplinary as well. You know, it covers biology and geology and evolution and a whole host of different elements. So yeah, it's really interesting to be able to bring them all together to try and solve these problems.
Well, thanks so much for speaking to me today and dishing the dirt on ancient soil. So I had to get a little pun in there. Yeah, thank you so much for being on Paleocast. And take care. Difficult times just now, so take care. And everybody who's listening, hopefully, will be inspired to get out and have a little look in the soil in their gardens, see if they can see what you've been talking about.
Yeah, that would be great.
Paleocast was brought to you by Dave Marshall and Liz Martin Silverstone, with contributions from Tom Fletcher, Vish Venkat, and Elsa Pancharoli. Music was composed by Patrick Kendall Smith. Paleocast was made possible by funding from the Paleontological Society and the Paleontological Association, but the show now relies on funding from you, the listeners. So, if you've liked this episode, please consider donating, and thanks to everyone that's helped out so far. Please visit paleocast.com for the supplementary material to this episode and for our archive of past programmes, and follow us across social media platforms to get all the latest news. Finally, if you enjoy our podcast, then please explore all of our video content on YouTube and follow our other projects, the Virtual Natural History Museum and the Paleocast Gaming Network.